Okay, hello everyone, and thank you for coming to the last think tank of the 2025-2026 academic year. Our speaker this week is Zachary Siders, an assistant professor with the University of Florida. Zach will be taking questions during his talk.
All right. Well, I'll take it away then. So, thanks, Trinity, for arranging this. Pleasure to be with you guys in your early morning out there. Um, I'm going to be talking to you today about some advancements to the good old classic von Bertalanffy growth model that I've been working on for a while. Kind of started in my PhD, and it's kind of continued ever since. So let's kick it off. So I'm sure most of you are well aware of the von Bertalanffy, maybe too aware at this point in your life, but it's a quick refresher. It's a growth model, one of the series that we use throughout fisheries that represents decreasing somatic growth investment as reproductive investment increases. And so we have this investment in somatic growth as a function of age. That's our kind of red line here down the lower left. And as maturity onsets, we get more investment into reproduction, and that growth, that somatic growth slows, and we have this nice asymptotic function that results. There's kind of two main formulations with a host of other variants out there. They all kind of stem from this top one, which is either this size at birth formulation, which has this L0 parameter in here. This is one that I use a lot in the work that I do because a lot of the things that I work with have viviparity. So they have a really high L0, high size at birth, and we have pretty good information on that. And so we'll talk a little bit about how we can bring that information into the model and inform that kind of y-intercept here, that L0. But the more classic version in fisheries in the lower right is our T0 version. This is a time intercept. It's one that was pretty commonly used because it allowed us to get rid of a parameter. We could set T0 equal to 0 for most teleos because their larval size is so small, and it would allow us to get rid of that parameter, which prior to kind of modern computing was pretty essential. T0 is really a nuisance parameter, though it can be an indication that you have size selectivity going on. So as T0 becomes more negative, it tends to indicate that we're not able to fit those early fish, those young fish. And so as a result, you tend to get this bias and this skew. And so one way that I look at T0 when I'm looking through a whole bunch of different growth models is I kind of look for those ones that are really, really negative. And unless that fish or that species has some sort of high size of birth, we can kind of assume that there's some size selectivity going on. But the VBGF really goes kind of all over the place in our fisheries management scheme and fisheries science. So we kind of start off with our VBGF. We can get a set of parameter values that come out of that. And those parameter values really nest their way into a whole variety of different parts that we're using within our management scheme. Some of them, like the most common one, would be using the VBGF to set our length at age schedule, or LT schedule there. And that, again, might lead to setting the weighted age schedule, because often we fit a length-weight relationship rather than a weighted age relationship. And so we have some dependency there that gets built in. Those then feed into things like yield per recruits, where we might use that in a really data-limited approach to kind of set risk, harvest limits or kind of risk envelopes that we want to look at, or for setting things like maximum sustainable yield at the end of a more structured assessment. The length at age can also lead to setting up things like the maturity schedules, and again, those really interact with yield per recruit, those harvest limits, those kind of fecundity schedules that we're interested in, and ultimately where those harvest schedules be for a lot of things like recreational fisheries, where we set length limits really based off of that maturity schedule. But we're also using a lot of the parameter estimates that come out of the VBGF for surrogate relationships, things like our really classic ratio of length of maturity to L infinity, or that asymptotic size. And this is kind of classically in teleos 2/3, and it's used quite frequently, particularly in data deficient species or species where we have a really hard time getting immature and mature fish to be able to set up maturity relationships. We'll apply those surrogates forward. So again, we kind of bake in the VBGF into another set of values that we're using. Really commonly now is using the VBGF or things like longevity to bake into the natural mortality surrogates. Again, it's a really common parameter that's really hard to measure. And so we're, again, building dependency on VBGF built into those surrogate relationships. And so you can both end up with issues where the surrogate relationships themselves are pulling in von Bertalanffy growth function parameters that might be estimated and might be biased as a result of size selectivity or other issues that result, as well as then in their application, we could be applying a biased VBGF and parameter estimates to a known surrogate relationship, and again, end up with some biased natural mortality, which again, those feed into setting harvest limits and things like that. An interesting one that we don't think too much about diagnostically, but is sigma here, the growth variability, and it can really tell us kind of what the validity is of setting up age length keys. If we have really high growth variability, then those age at length are really spread out. Something like here in the Gulf is Vermilion snapper, kind of famously just insane growth variability. And so using age length keys, a really common practice in our assessment approaches where we have a subset that are aged and a larger set with lengths, we can't really get back that age composition using an age length key because there's so much growth variability. So again, it provides this kind of diagnostic of kind of the use of age length keys there. So VBGF, it kind of goes everywhere. Again, other growth models have similar kind of structure and how they play with these other kind of outputs. But really, VBGF is the most common one. And so one of the advancements that we've been really pushing in a long series of kind of case studies is using Bayesian statistics. The background here is, originally we were estimating the von Bertalanffy kind of graphically, and then we moved over to most commonly using nonlinear least squares, and that doesn't provide us with that sigma parameter, which can be quite useful to think about and utilize. We've now kind of moved over to maximum likelihood estimation, and the last kind of decade or so, we've started seeing a lot of Bayesian statistical applications applied. And so I'm sure most of you are, again, familiar with Bayesian statistics, but just a quick refresher here. When we're traditionally using maximum likelihood estimation, we're asking the question of how probable is the data, given that our model is true. One of the challenges there is that, particularly in something like the VBGF, we might not know what the model is true. We have a kind of set model structure, but whether we share parameters between sexes or between sites or something like that, we again might not know what that model structure is true. So we kind of end up with some other model comparison techniques that we commonly use to sort our way out in that maximum likelihood framework. But the posterior in the Bayesian statistics kind of flips it on its head and asks, how observable is our model given the observed data? And this is quite useful because it's kind of the inference, kind of native inference for humans, but also kind of the inference engine that we're looking for when we're building kind of these classic von Bertalanffys, where we want to kind of know, are parameters shared or are they not? Can we even estimate these parameters? How sensitive are they to prior information? Those sort of things. We can ask those questions within the Bayesian statistical framework. To do so, though, we need to bring in prior information. Yeah, go ahead.
Zach, sorry, we have a question online. Andre, if you would like to unmute yourself.
Sure.
Yeah, could you go back one slide? I just want to
Sure.
understand when you say that age length keys are biased when sigma is high. I don't think that's true. At least that hasn't been true for the theory of age length keys. In fact, the reason you have an age length key is because there is variability in length at age.
Yeah, I didn't. Yeah, I wasn't.
Bias is a very strong term, particularly when it comes to things like growth curves.
Yeah,
and I'm not sure I'd use that term.
Sure, I think you're 100% right. Bias isn't exactly what I meant there. It meant that the validity of using them comes about when we have really, really high growth variability. We end up with really smearing a lot of ages across a lot of length bins, and so as a result, they end up being, our age compositions get really tricky to pull forward. And we end up having something like Vermilion here in the Gulf, we end up having to kind of projection age all those fish because we can't really use the age length keys in that instance. But yeah, you're right. They're not, bias is the incorrect term.
Okay, that sort of helps. I don't think that's actually true that you have to age individual fish, but you need large sample size, clearly.
Sure.
Right, let's see where we go. Thanks.
Okay. So the returning to our kind of Bayesian statistical framework here, when we're talking about the, we can add in that prior information, and that provides an opportunity for us where we can kind of bring in information that we otherwise might already know and might have really good ideas about. So things like that size at birth relationship or a parameter estimate, we might have a really good idea of what that parameter is, and we can bring that into the model and use that, as well as capture the uncertainty in it using a prior. The marginal, again, is asking how probable is the new data under all possible models, and this is where we kind of brute force our way through with things like JAGS or STAN or other ways of estimating those Bayesian models. So what we're going to do next is we're going to kind of walk through a bunch of case studies where we've taken this Bayesian statistical framework, we've applied it, and kind of building in some of the different ways that we're handling some of the issues around building these von Bertalanffy growth models. So one of them that's a pretty natural extension is, if I can build a von Bertalanffy for one dimension, can I build it for multiple? And so we started off way back in my postdoc looking at Brazilian electric ray with some folks in Brazil, Fernando Rolim. And one of the questions that we had when we were building this model was kind of a really classic one of saying, well, when we build these models, are the parameters like our growth rate here, our Brody growth coefficient, is it shared between sexes or is it sex specific? And so when we build this model originally, we built it for total length, and we see that K is really not that different between them. So that the individuals are growing to different lengths, but the kind of rate at which they reach their maximum lengths is roughly the same. On the other hand, when we build in things like our total disk width, which is really important for array, it brings all these kind of other physiological, biological, and ecological processes of, one, it generates lift, so it reduces kind of locomotory cost. The bigger you are, especially in multiple dimensions, the harder it is for a predator to eat you. And in particularly in the Brazilian electric ray, they're electric ray, and so actually the bigger you are, the kind of bigger electric field that you can generate to deter predators as well. And so there's some really good kind of correlates here that go along with length. And so if we were only to fit total length, which is kind of the standard that we would do for most fishes, we would actually conclude that there's really not sexual dimorphism around this kind of growth rate here, when in reality there is. And what we see is that males are generally sleeker, they're investing more in total length and being able to move around, versus the females are much flatter. And we again think that probably has to do with, predation, being able to survive, to reproduce, as well as being much heavier, carrying all those young. It allows them to generate a lot more lift and probably reduce their locomotory cost, as well as just be larger to produce more young. And so the next, if we can do it in one dimension, the obvious thing is to say, well, can we do this in even more dimensions? And so in this case, we have a case study where we just recently published this on Florida soft shells with the Apollon ferox. And we measured carapace length, plastron length, that's the underside of the turtle, carapace width, and shell height. This is all through mark recapture data. And the interesting thing for the mark recap process here is this is all done by hand by snorkelers in Florida Springs, and they're very picturesque and nice to catch turtles in. But you're capturing this turtle with ferox as its species name, which is kind of ferocious, nasty turtles. They're big claws. They tend to fight back when you try to capture them. So not a lot has been done on these, and they've been kind of depauperate on what we know about their life history. But they're also a soft shell turtle, so we can't traditionally mark them where we do kind of engravings into the hard carapace for turtles, the hard shell. So instead we tattoo them, which is what you see in some of these photos here. And this work was done by the Turtle Survive Alliance and a collaboration between us and them. And so one of the things that we see, which is quite fascinating when we start looking at these multiple dimensions, is we start to see where investment is really happening in somatic growth and where those trade-offs start to occur as individuals mature. And so one of the interesting things that we see here is that sexual dimorphism in terms of size really depends on when you're looking at in the lifespan. So When we're at young ages here in this top row, these are the males in the shaded area. This is their carapace width in the shaded, and the line is their plastron length, and the width is obviously carapace width. And these little humps are their shell height. You can see that the males are bigger than the little unshaded circle in the middle, which is the equivalent female on the bottom. And so males are actually larger than females, really up till age 15, where they're about the same size. You can see that here. And then females continue to invest into somatic growth long after males, and eventually get much, much bigger than males. And so when we go out in the environment and we kind of think, well, what's the sexual dimorphism out there in the environment? It might be that most of the turtles females are larger than males, given kind of the population structure. But in terms of the lifespan, it is not quite the equivalent. The other interesting thing is that we see females are kind of slightly more domed. Their shell height is getting much bigger. Their carapace length is getting much longer. They're getting a little bit wider. They're mostly invested in length. And if you ever seen one of these turtles swim, they kind of swim with an upward turn angle, and that again generates a lot of lift on the underside of the turtles, which if you're carrying a big clutch of eggs multiple times a year, that allows you to carry that extra weight, generate locomotion, as well as avoid predators. Most of the males we basically see most of them are being eaten by alligators by the time they're age 20 or so. It's quite interesting. So we've kind of talked about dealing with kind of multiple dimensions and what that can tell us and lead us to some interesting information about kind of the biology and ecology of those species. But one of the common things that we have, particularly in teleosts, is we have aging error associated with aging those hard parts, whether otoliths or vertebrae or spines. And so particularly for you folks out there, you typically have a lot of production aging labs, and they're aging lots and lots of otoliths with lots and lots of readers of them. And as a result, you have all this extra information that's coming in from those individual reads. Occasionally, we apply some consensus aging scheme as a result, where we take something like a mean, median, mode, or other kind of process. We might kind of revisit reads that we think are biased, and we kind of go back into that process. But this consensus aging really ignores the fact that we have this really rich observational data set on the multiple readers. And so even with the kind of unbiased read, so all the readers are kind of committing aging error randomly, we end up with consensus ages that can lead us to more certain VGBF parameter values than actual reality, because we haven't captured all that aging error forward. And kind of classically, when we compare readers, we often see this pattern below, where we have increasing error with age. So not only are we making kind of more certainty about the parameter estimates, but across ages, we're also ending up with more uncertainty in terms of how we're reading those individuals and not carrying that forward. And a Bayesian allows us to easily bring in that data and build some hierarchical models to handle this information. Consensus aging can also lead to biases depending on how it's implemented. So if our kind of example here, where we have a true age is 5 and our reader 2 is getting a 6, and then we take the mean age, we get an age of 5.33. And that would be the age that we then build some classic length at age von Bertalanffy model for. And so if the reader biases are non-random, though, we can lead to issues where we have kind of consistent underaging, the length at age is lower than true. This leads to higher K values, which again, if we pass it through something like a natural mortality surrogate, we end up with higher M values, and we make different conclusions about the sustainability of that stock to fishing. And vice versa for overaging, where we can end up with lower K values and lower M's, and again, assume that stock is less resilient to fishing and harvest. And so it's really important that we think about how we deal with it. And again, the Bayesian way allows us to basically just say, well, let's use the raw data. And so one of the things that we commonly report out of here is this index of average percent error, but it's really not a measure of kind of the bias of the reads, but more the between reader error, and that was what it was designed for. And so we often need to validate ages to measure this bias. And so for something like Derek Chamberlain's work on black-bellied rosefish as part of his dissertation, he's now at Alaska Fisheries Science Center, we validated that aging techniques on black-bellied rosefish with bomb radiocarbon, and that's a whole other set of work that we're up to. But what we see is that this error is often pretty minor with short-lived species, but for something like black bellies, which are the rockfish of the Gulf here, they're long-lived, they're late maturing, they're slow growing, and they're really hard to age. They can live up to 90 or so. We have really, really tight opaque zones there, and it can be really challenging for people to age them accurately. And so as a result, we want to be able to bring in any of that aging error forward into any von Bertalanffy that we build off of this. And so in this case, this was done for Derek's dissertation, and so we didn't have some production aging lab to go off and make 6 or so reads of these fish. We had 2 readers, and each dot here on this von Bertalanffy function is a read, and we have a bar that links the pair of them. And so our max fish here out at the end was either 90 or 94 years old, depending on the reader. And we can see that looking at the distribution of the difference between the 2 readers here, most of our reads are centered around 0 and plus or minus 5, which isn't bad for a fish that lives this old. But we do have a few here that are minus 15 or plus 12 that are concerning, and we can kind of see them in the graph. We have some really long bars linking the 2 of them. And so again, this is a place where the Bayesian von Bertalanffy can allow us to bring that data in. So how do we do that? So what we did was we, again, assumed that those reads are data that inform the observation process of the true age, and that true age is treated as an unknown and estimated within the model. And so we set up a generalized t distribution here where we have t sub I sub r, which is our observations of age, our reads, and then we have our number of reads, which inform that degrees of freedom new parameter. We have our unobserved true age that we're estimating within the model, and we can pull out that observation error that comes from that aging error. So we can directly estimate it in the model and separate it out from the error that is from growth variability. And so we bring that over, we get that, we estimate that within the model with our hierarchical process. We pass over that unobserved latent true age into the von Bertalanffy kind of classic size of birth model here. And we get estimates out of the back end of them. And again, we can separate out those error processes, the growth error, which is what you're seeing in the shaded gray region here, from the observation error. Yeah, Andre.
Yeah, quick question. Yeah, so you still, I haven't read the paper, which I should have, but you're treating the mean as the data rather than the individual reads as data?
No, we're treating the individual reads as data.
So then why is the number of reads the overdispersion parameter, or I'm misinterpreting the generalized t? Because why would having more reads change the overdispersion in the individual reads? Certainly in the mean, that's true, but not the individuals. What was the logic there?
In this case, so I is the
animal.
So, yeah, I is the animal, R is the reads, and then we're estimating that latent age for the animal, and then
Right.
we're sharing the overdispersion across all animals.
Well, I'm just looking at this. V is the number of reads, which usually in a T distribution is the overdispersion, is the degrees of freedom.
Yep.
Can you explain that? Because you've got individuals here. Why would having more reads change the degrees of freedom? Because you're fitting to the individual data points.
Yeah, I think what we're assuming here is that we have that as we get more reads, we're going to be more certain about the individual's unobserved true age. So that's why we're using the number of reads as the degrees of freedom here.
But in this case, it was V equals 2 anyway, so probably wouldn't matter too much. But
no, it doesn't. But as we get more reads, it matters.
Yeah, I'm not completely sure that's the way I do it, but yeah, okay. I can see what you did here. Certainly the T is quite cool.
Yeah, I know. I'd love to chat more about it if you want. It'd be great. Love to hear what you're thinking. Oh, so yeah, so to close this one out, we have our multiple readers here, and we can pass that information forward. Not shown here is that we have, we've kind of separated that error out. So when we get those parameter estimates out, we've dealt with that observation process, and then we can use that forward. And in the case of our black bellies here, you know, they're growing pretty slowly, and their natural mortality is very low. So they're likely pretty unresilient to fishing. Luckily for them, they don't really have much of a directed harvest. It's mostly incidental bycatch, but certainly something we're kind of keeping an eye out for, and we're currently looking at some stuff in the Atlantic for these ones that have a more of a take from a deep water grouper harvest. We can also start looking at case studies where we have basically the absolute worst scenario where we have like n's of 1. This is purposely was done as kind of a cheeky study to see how much information we could bring in that would help us inform these models. And so this is again out of Fabio Catavallada's time in Brazil when he was doing his PhD. There was a goblin shark that was caught as bycatch, and we had a single vertebrae to age. And so again, we're like trying to see if we can push the bounds of how this works and get an idea, and how do we work our way forward for these like really rare things where we're going to have really, really low sample sizes. And so this is a deep water shark. The vertebrae are basically like jello. Fabio had this kind of light bulb moment to stain it with Alcium blue and was able to resolve out some of those banding patterns. And then we were able to measure and make some increments off of those and get increment analysis that we're able to measure those and move those forward and bring them into the model. But we have one shark, so we need to bring in a lot of information to be able to help us inform what those parameter values might be for the Bertalanffy. And so in this case, we have this great data set compiled by Rigby and Simpfendorfer on shark life history traits and also some of their habitat and reproductive mode. And so we built some really simple regressions off of them, looking at the relationship between size of birth, the Brody growth coefficient, and age of maturity as a function of their maximum size. We were able to then go get maximum size from the literature of goblin shark males, which is the male that we had, and we're able to apply it into this model, assuming that they're a deep shark and that they typically have an adelphophagic reproductive mode. And so these life history correlate approach that comes from the Rigby and Simpfendorfer allows us to build informed priors for the Bayesian modeling. And so each one of these panels shows the relationship between K, age, maturity, and size at birth for that maximum size for males, and the little crosshair is where goblin sharks fall out. But even then, we still don't really have great data on things like what the maximum size is. We had a 27-year-old, and so we assumed that probably wasn't the maximum age these sharks can reach, given what other lamniforms do. And so we went off into the literature and grabbed all these maximum size records, as well as a bunch from fishing magazines. There's a pretty cool one. I think it was out of the Murray River, where it was, some guy basically being like, look, I caught this crazy big goblin shark when I was off fishing. And so we were able to pull some of that information forward and bring that into the model as data on informing the maximum size. And then we had a couple reported records of the smallest free swimming individuals, and that brought in 2 data points for L naught. And again, we're kind of pushing the absolute bounds here of how this could work, but the point is to say, can we bring in some of this prior information, bring in our N of 1 increment data here that informs a latent length variable, and use that to get an idea of what the preliminary growth might look for this species. And so we bring all that informed prior information in. The kind of shaded area is the posterior on each one of these, and the solid line is the informed prior that came out of that Rigby and Simpfendorfer approach. And then we have our increment data here in the dotted lines with the bar around them is the uncertainty. And the reason why there's uncertainty in them is because in this approach, we're using a back calculation approach from the increment data. And that back calculation typically depends on size at birth. And so in this case, we're doing the back calculation and estimating all the VBGF all at the same time within the model, and passing the uncertainty in the size at birth into the back calculation, which is why you see that error kind of increases as we get closer to the size of birth because we're more and more uncertain about what the length at age would be in the past, and we have a known length at age for the capture size. And so it's a cool approach. We tried it here. We'll talk a little bit more about it here in a little bit on some sawfish. So up till now, other than kind of our soft shells, we've been talking about being able to age individuals. But for a lot of things, where aging is sometimes not feasible. In the case of a lot of hard-shelled turtles, they have reabsorption in their humeri, which is kind of the traditional hard part that we use to age them. And so we have all these issues with trying to, back calculate how many rings were lost in the humeri, and it becomes quite tricky and pretty error prone. And so typically we use is mark recapture. Also, people tend to frown upon you killing a bunch of turtles, and so we typically want to do some sort of non-destructive technique. And so in the von Bertalanffy, we use the kind of Fabens approach here, which conditions the von Bertalanffy growth model based off of the capture and the next time, the capture that we've observed now, and the change in time between them. And we can build that model out a little bit more by dealing with the repeated measures that we get from the mark recaptures. And we need to be able to do that because otherwise individuals that had tons and tons of marks would kind of unduly weight the model in their direction. And so what we do is we build in individual random effects. And in the case of the model, we can actually structure that individual L infinity or K outward, where we can have a shared kind of intercept L infinity that's shared across individuals. We can include some fixed effects, and we can structure that however we want. In the case of this, we included some cohort effects, some environmental effects, as well as the spring heads where we were tagging these turtles are heavily used by humans. So we included some human use effects on there as well. And then we can include random effects. And because the von Bertalanffy, the K and the L infinity parameters are so tightly correlated with one another, we wanted to draw these random effects from a multivariate distribution that deals with that covariance between those parameters. And so we have over here in a little box, we have our vector of random effects for L infinity and K. They come from a multivariate normal distribution, which is centered on 0 and has a covariance. And then we set that covariance up with the Koleski decomposition for it. And this allows us to, again, estimate these models and bring in those random effects and deal with the repeated measures, as well as any of the issues that result from individuals being larger than L infinity in their mark recapture time series. So when we do this, we see some interesting things that come out for these turtles in these springs. We can look at things like differences in the asymptotic length, obviously. We can look at how those growth trajectories change over time. In the case of our biggest turtle, our Swaniensis here, these are like big monster turtles that river cooters you would see in the spring heads. They grow about the same between males and females up till about age 8. And then males kind of quickly reach their asymptotic size and females continue on. And the important thing here is they're a hard-shelled turtle, so there's a really strict limit on their reproductive output based off of how big they are. They can only fit so many eggs in that hard shell. They have no room to really expand. And so we typically see turtles, females, continually kind of invest in somatic growth, even though their reproductive age, so they continue to increase their reproductive output. What's cool about this system is that these are springs. They're thermally stable. They have really rich aquatic habitat in them. So for being a herbivorous turtle, these things get monster in the springs. And in fact, when we look at our Pseudomys texana here, we see that they're getting really, really big relative to their non-spring sites, which is in the green next to them. And so we're getting really big turtles in these spring heads, and we're able to pull all that out and deal with all the repeated measures. And some of these turtles were captured, like 39 times over the 20 years. And then we have some turtles, were captured once or twice. And so we can deal with all of that within the model and the repeated measures component. Just to kind of put a fine point on how big these turtles are getting and how fast they're growing is we're going from our little hatchling here to a year 1 juvenile, and they're putting on about 20% of their maximum size in that first year. And so they're growing super, super fast in these springs. And it suggests to us that these springheads are really kind of these bastions of abundance and reproductive output, and might be a good source in the larger environment. And this is kind of pretty remarkable given that they have a hard shell and they're having to do all that skeletal restructuring as they're growing. One of the other things that we typically run into is that we have alternative structures that we want to go about. So in the case of the gray triggerfish here, we have the motivation is that, this is again Derek Chamberlain's work from his dissertation. You have otoliths that are super tiny. You can see the little sagitta otolith in this CT scan here over on the left. We have these incredibly weird, like multi-dimensional otoliths with all these like little frills on them over on the right. And these are really, really fragile, and they're really, really weird, and it's hard to like read them at all. And this is like the antithesis of what we would want if we were to do production aging, something that's hard to get out, hard not to break, and really hard to read. And so the motivation was to swap over to dorsal spines. The way that that technique had been done was that there was pretty high index of average percent error between readers, and with some OTC marking and some bomb carbon validation, bomb radiocarbon validation, we were able to see that the old way of measuring spines was underaging by several years. And in the case of gray triggerfish, the kind of maximum size that we're seeing in the fishery, maximum age that we were seeing in the fishery historically was kind of age 10 or 12. And then that got bumped up to like age 15 or so with a new spine method that was developed after this kind of OTC marking technique. And so that really shifts the age composition, really shifts our understanding about their life history as a result. And now we kind of have to go back and remeasure all these spines that were done with the old method to be able to update our age comps moving forward. And you can see this otolith down here in the lower right that shows those really tiny little structures that we're measuring for age here. So again, it's really, really tough to age them. But one of the questions that we have is kind of, how different are these different techniques? We have otoliths, we have new spine, we have old spine. How different are they? What are they doing? How are they showing up in the von Bertalanffy, and what are they doing to our understanding of those structures and schedules? And so we have the same Brody coefficient, the big K value that we're really interested in for otolith on the left, new spine in the middle, and old spine on the right. And really quickly, you can just immediately, looking at those posteriors, see that our old spine is estimating a much higher K value. And again, we know that that's the case because we've OTC marked them and shown that they're underaging as a result. And so we would make really different conclusions about those kind of length at age schedules, when fish are reaching those lengths, and the resilience of those stocks given this old method. When we look at the new spine, we bump that K value quite a bit down. And for the most part, the posterior parameter estimates for these two methods generally overlap. Things like sigma are really similar. We get a similar male K rate, K value between the two methods, but the otolith is estimating a slightly higher K value for females. And there's fewer otoliths here. Again, when we did all of this, though, we had multiple different methods we want to be able to estimate all of these things in one model. So we're fitting multiple von Bertalanffys to each of these methods all in one go so that we can get posterior parameter estimates on the back end that are all drawn from the same sampler. So then we can kind of compare each pair of posterior parameter estimates to one another to really strictly understand where are things different, how are they different, and to what degree. The interesting thing that's happening in the background of these is also we're dealing with that multiple reader issue. We have 3 readers for each one of these. And so in this model, we're also dealing with the fact that we have multiple readers for otoliths and new spines and bringing and separating out that aging error in that process. And this kind of larger being able to compare parameter estimates is something that we really frequently want to do. And the old way that we've been doing it with maximum likelihood is we typically kind of run a bunch of different models, and then we use something like AIC model comparison on the back end to say which parameters are shared and which ones aren't. and we're kind of bootstrapping, pulling out of the back end of these AICs some kind of guesses of what is going on in terms of the biology. And typically you kind of see folks either making really explicit kind of multiple hypotheses and comparing them with AIC, or kind of the alternative, and less frequently you see kind of dredging, where they kind of run all possible models and see how these things shake out. And so over on the right is our example from our mark recapture data on our pseudomaize, those 4 different species, and we have our different parameters of L infinity, K, sigma, the change in the growth over the first year, and age of maturity. That was pulled from a surrogate value. In the Bayesian world, we can just estimate the full model, and then we can look at and measure the overlap between them, and we can see exactly what parameters are different, how different they are, and if we want, we can do some significant testing as well as in that vein. And so in this case, we can see things like our pseudomaize nelsoni, our Florida red belly turtles. They're pretty overlapped for L infinity and K, and so we would probably conclude that they're not statistically significantly different between one another, versus something like our Peninsularis and our Swaniensis. We have pretty minimum posterior overlap, and we think that there's probably some size sexual dimorphism going on as a result. Okay, so we talked about a bunch of case studies. I'm going to talk about one. Yeah, Andre, go ahead.
Yeah, just wondering if you've tried Bayesian model selection along with I mean, overlap makes sense until you start to think about the multi-dimensional surfaces here, because you're showing marginals, not the whole posterior. Have you tried Lu and those kind of things, which
Absolutely.
Get spat out? Did you get anything interesting?
Yeah, I mean, you can get comparative results to the AIC model results for sure. I think in the case of this one, we did the Lu-IC model comparison. I think we see basically what I just was mentioning, that the Peninsularis and the Swaniensis should have separate parameter estimates versus the Nelsoni. I think maybe Texana too was also separate sex-specific parameters.
Thanks.
Yeah, so we've talked about a bunch of case studies here, and now we kind of want to talk about bringing it all together here. And the challenge and the motivation for me is that a lot of the species that I work with are typically protected species. We're not going to get a bunch of the kind of data-rich data sets that are useful for data-rich assessments. And so we're typically ending up these places where we're making pretty simple stock assessments, or doing something like population viability analysis to kind of game out what this is going to look like in the future. And again, we're making a lot of kind of simplifying assumptions and being able to do those sort of models. The reality of it is that once we're kind of in that space, most of the time we end up with data deficiency kind of throughout a lot of our life history, and things start relying really intensely on growth. So if we're kind of lacking a bunch of these other data sets, we typically have pretty uncertain maturity schedules, and so we might end up using something like a maturity surrogate. We're almost assuredly missing something like natural mortality, unless somehow we get a bunch of money to do something like a tag experiment or a telemetry experiment and measure that. But again, that can be difficult, and often small sample sizes and small pools of species, and so we kind of are concerned about using those. So again, we kind of rely on surrogates here. And all of that, again, that information feeds forward into things like yield per recruit analyses and harvest limits or population viability analyses. And so again, we kind of end up putting a lot of weight on the growth, and we really want to make sure that we're doing it well and kind of dealing with our sources of error and other sources of bringing all the data that we possibly can forward. And so the example here is small tooth sawfish. This was the first marine fish listed as endangered under the Endangered Species Act. It was listed in 2003. They're a shallow water, generally shallow water species around the state, pretty charismatic. There's some illegal trade going on for their rostrums. So we typically get ones that have their rostrums cut off, which proves to be an issue in terms of building some of these models when we want total length. And so we also had the issue of data deficiency. I think the archival sample size was 96, which is not absolutely terrible, but it isn't great for building a kind of traditional length at age von Bertalanffy model off of. We're likely to have some size selectivity biases going on and just a lot of uncertainty in those ages. And so we kind of took a bunch of the different approaches that we've kind of talked about here and pooled them all together. So we have length at age data coming from those kind of archival samples. We had a mass mortality event that we'll talk about here in a sec that we were able to get more vertebrae from and get an increment analysis on to really bump up our idea of what the growth trajectories look like. We had some mark recapture data where we had like 1 or 2 recaps per individual that can be used in the model. So again, not enough really for us to be able to build our own mark recapture model because we don't have enough recaptures over time. But in concert, we can kind of use all this data together. But it introduced a new challenge that was a kind of a nice way to bring in and handle some of the uncertainty that can result. In this case, uncertainty in length at age. And so in this case, we have individuals who are coming back as carcasses for the most part. And so their rostrums are typically removed, the people come across them. So we have a whole bunch of individuals where we don't have their total length, including the rostrum. And then we also have carcasses that have been floating out there for a while and they get eaten by sharks. And so we might be missing the tail of the sawfish. Then we don't have, again, the total length. And so we wanted to be able to build a model that estimated these length-weight relationships within the model itself while passing all that error forward in terms of the length at age uncertainty. And so you can see from these 2 relationships, these were estimated within the von Bertalanffy growth model alongside everything else. And you can see we have pretty tight relationship, very little error in body length and stretch total length. So if we're just missing the rostrum, we're pretty good. If we're missing part of the body, usually the tail because the shark ate it, we have a lot more uncertainty in using the rostrum length and extrapolating to the total length. So again, we wanted to capture that uncertainty moving forward. We also wanted to bump up all those archival samples using increment analysis. We kind of knew we had this approach from goblin sharks sitting out there previously. We had this mass mortality event, and one of the students in one of my classes working on this as part of their thesis, and they basically disappeared from class to go deal with this mass mortality event. It was very much all hands on deck. It was really large sawfish, as you can see in these photos. These were often reproductive females, and we didn't really quite know what was going on, but we were getting a lot of them really quickly. And for something that is endangered under the ESA, this was really, really, really concerning. And so I encouraged them while they were there to just get as many vertebrae as we can so we can be able to do the increment approach that we did. And so how this works is it's pretty simple, but basically what we do is we have this vertebral section here shown on the right, and we can get a birthmark out of it, which is where this 0 in red is shown. We can measure those birth bands moving forward. This is an incredibly easy sawfish to read. I assure you, most of them were not this easy. They're much harder. And then we have our edge after our age 4 band, and that gives us kind of our 4+ our leading edge. And that's where we captured this individual at 300 or so centimeters. And so we're basically trying to back calculate between the capture size and the burst size. But again, we're estimating that burst size within the model. And so we want to pass any uncertainty in the burst size into that back calculated length at age. And so what we do is we measure the increment distance, where we're just literally clicking all these bands and measuring them in pixels. We then convert those pixels into a relative increment based off of the edge and the birth band. And then we can use that relative increment in our latent or in our back calculation. And in this case, we're doing that back calculation within the model. So we're reading in those increment data into the model themselves, doing the back calculation within that model and getting this latent length at age. That gets calculated within the model and then used in our kind of classic size at birth von Bertalanffy moving forward. And the great thing about the increment analysis is that, again, gives us this really rich data set of giving us these growth trajectories. They're hyper informative to the model as a result. But again, we have repeated measures per individuals, and so we need to deal with those individual random effects. And so we borrow that similar approach that we were talking about for the mark recapture, and we can use that here for individual random effects on L infinity and K to handle those repeated measures. And so in the lower left, you can see some different different vertebral sections here with one of our older individuals here getting really stacked up towards that edge and quite challenging. The other thing that these sawfish do is they move a lot back and forth between estuaries and spring heads and mangrove habitats in and out. And so as a result, being in kind of a tropical environment as well, we can get a lot of kind of false banding and things like that, which can make this slightly more challenging, but we're able to work our way through it and get some consensus increments. We can also bring in a bunch of mark recapture data. As I mentioned, we had kind of usually at least kind of 2 time points, if not 2 or 3 per individual, but we weren't looking at really long mark recapture trajectories. These individuals will move between the Keys and the Bahamas, and so this is kind of a big joint mark recapture data pooling exercise between the 2 different nations to put it together and be able to estimate what's happening with sawfish. And we use the kind of pseudomaze turtle approach as within the model itself. And so just again, to really reemphasize, we're doing all of this within the model. So we're doing all these different, bringing all this different information together, all within the same model itself. We're able to start with things like our lengths down here on the lower left. We can filter those and make sure that the ones that have lengths or those lengths are being estimated and that error is being passed forward into our length at age model. That length at age model can inform parameter estimates like L infinity, K, and L naught. We can also bring in direct observations of size at birth, which were occurring from some fisheries independent monitoring down in the Florida Everglades. And we can use those size at birth observations and bring them in as independent data into the model. We can bring in that mark recapture data and use that Fabens von Bertalanffy to inform L infinity and K, but it doesn't tell us anything about L naught. And then we can use our increment analysis along with our estimated L naught within the model to do our back calculation, which again feeds into that length at age model, which feeds itself back in. So it really is a big data pooling process here, and really a nice way for something that's so data deficient as sawfish to be able to pull all that stuff together. You can kind of see the effects of some of that length extrapolation here, where anything that has like an X or a plus was a individual that had a partial length. The kind of pluses are all body only carcasses, so that was one that we had more certainty in the length extrapolation. And then we have ones that were rostrum onlys, and you can see that vertical bar around them is giving us a much wider band of the estimated length at age from those length extrapolations. And along the way, we can do everything that we've already been talking about. We can compare parameter estimates. We can estimate the full model and compare sex-specific ones. In this case, to Andre's question here, the LUIC says that there's a shared parameter estimates between them. So there's no sex-specific differences in growth, which is pretty cool. So in conclusion, Bayesian methods offer us a lot of clear ways we can to improve our von Bertalanffys. We can inform prior parameters with prior knowledge, like we did with goblin sharks and some of the other ones, bringing in some information about size at birth. We can estimate growth in multiple dimensions and the correlation or covariance between them. We can handle uncertainty in age and lengths within the model itself. We can handle repeated measures. That comes about things like increment analysis or mark recapture. We can bring in multiple data sets. Again, we can kind of make the most use of what we know and what we have available to us, as opposed to kind of just using a traditional length at age and kind of leaving all these other data sets and information on the table. We can do hypothesis testing. It's generally way easier than MLE because we can get those posterior overlaps and really look explicitly what parameters are different. And then most importantly, we can pass all that uncertainty on to any derived values. And we can just do that derivation within the model itself, very similar to what we do in a lot of the stock assessment framework, and we can just pass that on outward. This was built through a lot of collaboration and case studies. As I mentioned at the top, we started off in my PhD talking with Brian Mathias, and we were working on some black crappie growth and kind of talking about how we do this. And we were kind of mucking about with JAGs of how do we build an environment and cohort effects. Fabio came to UF and did his postdoc here with me as postdocs together, and we worked on a bunch of stuff on guitarfish and electric rays and goblin sharks. Through some of my sea turtle work, I got hooked up with some of the freshwater turtle folks, and we've been mucking about in that space for a while now. And then I was fortunate to be part of Derek Chamberlain's graduate committee and got to be involved in some of the work that he did here. But there's my contact information. Happy to take any outstanding questions. Thanks for your attention. Yeah, Andre.
Well, first, anybody else? I've been asking too many questions, but if anybody else, oh, Terence first, and then I'll come back later.
Cool, Terence.
I think Terence was clapping. I don't think he had a question.
Okay.
Oh, I do. I was wondering, to what extent have you, and I've always wanted to do this and I've never done it, but have you taken these posteriors a little beyond the M and TM into things like reference points and stock status, just to see to what extent the uncertainty is actually captured by it? I mean, it's it's doable. I haven't done it yet. What's your experience with that?
Yeah, we haven't published anything on it yet. Where it's kind of in the works. We've been messing around with just really simple yield per recruit analyses and passing all this stuff forward. And it kind of depends on where that uncertainty comes in. So if you kind of, in some places, we can kind of pull out the growth error, right, the aging error, and we can separate it out from the growth variability. So we actually kind of get typically less uncertainty in the reference points in that case. But in other instances, when we're kind of capturing all this error that's coming in, we can also end up with those reference points being much more uncertain as a result. It kind of depends at the moment, but it's certainly something we're interested in playing around with. Fabio messed around with some of that when he was with Will White at University of Oregon, or Oregon State, sorry. Kelly, I think.
Yeah, thanks. That was a great talk. I have a question. Well, a little bit of background. Excuse me. I'm the project lead for the Fisheries Integrated Modeling System, a stock assessment framework that we're trying to get up and going within NOAA Fisheries. And we currently have a contractor that's doing some really great work to internally estimate growth, because right now we just use empirical weighted age instead of estimating growth. And we can allow, our input data structure allows for in the future having individual records in there, but obviously now we just have the compositions. Mm-hmm. And so I'm kind of wondering this trade-off between estimating growth externally and then potentially getting estimates of uncertainty of those parameters and being able to put that into an assessment model as an external estimate, but an uncertain estimate, mm-hmm. versus muddying up, potentially these large assessment models that already fit to a ton of data and have a ton of parameters, and potentially putting this framework into an assessment model, and the pros and cons of both approaches.
Sure. I mean, I would say, at least in my own experience, like, we've been building these kind of bespoke, and there's kind of intricacies of each one of them that we've kind of had to work our way through. I think there is probably opportunities to kind of, and we're working on an R package, but I haven't gone through tenure yet, so my attention is spread quite thin. But we're trying to put an R package that builds all these things together and kind of makes it a little bit more unified ability to kind of click your way through what options you want and build a model out of the back end out of it. The challenge, though, is you have kind of enough bespoke things that would result, I think, in most of these things that they probably wouldn't end up in an integrated stock assessment to begin with for a lot of these species. But certainly something like the multiple reader approach it's pretty fast, and I don't think it would really cost much to be able to do it. Now, I think Andre has some points that we probably could think about what that distribution looks like for the multiple readers, but I think that's probably the best bet and probably the most useful frame for an integrated stock assessment would be pull that one out and use that one. Does that answer your question, or a sufficient dodge?
Yeah, that's great.
Okay.
Regarding what parts to potentially bring into an assessment. What about the second question that was in there about just externally estimating growth and putting those in as parameters into a model? Because something like this, where you said like each one is slightly bespoke and you're able to do things that you really can't do in a canned assessment framework, is it better, or what are the repercussions of just estimating it externally and then putting growth parameters into the assessment model?
I would say the hard part is like dealing, being able to read in this particular instance, the joint distribution, right? Because you have covariance between those parameters. You have actually quite a bit of covariance between, particularly L infinity and K. And so you want to be able to pass that uncertainty forward into the assessment in that manner. So if you have the ability to do that, then it's relatively straightforward. Estimate the DBGF, deal with all the intricacies that you need to, and then you can pass that joint distribution forward. If that's not built or the mechanics aren't there, then I think it becomes quite tricky to deal with that forward.
Yeah, thanks. I'm a little bit sad that Andrea Haveron isn't on the call today because she's working on multivariate priors and the ability to pass these correlation matrices and such.
Awesome.
And so my naive understanding of it is that in the future we will have the ability to do that. But today, Adrian, who's on the call as well, is working on just implementing the very basic von Bertalanffy. But we'd love to chat in the future and potentially try to work this out.
Please, yeah, absolutely. Yeah, Andre.
Yeah, just to emphasize that if you are going to bring priors in, really jumping into the multivariate, because these things are very highly correlated in some cases. Not the sigmas probably, but certainly L infinity and kappa. In principle, your models all parameterize in terms of L infinity and kappa, but you can reparameterize them in terms of things that actually are less correlated, which is why we, that parameterization we use like in stock synthesis.
Yep.
That might allow you to get away with uncorrelated inputs. But yeah, if you're going to use L infinity and kappa and you don't have data at either top and bottom end of the length age distribution, yeah, you've got to do it properly. Otherwise, you can go way wrong.
Yeah, that was kind of part of where Fabio and I at one point we come out of doing a bunch of this ELAZABranch stuff, and we were kind of just trying to get kind of a snapshot of what was happening out there in the literature of how these models were being used in sharks and rays. And we just were like, wow, there's so many of them that have so much size selective bias built into these things that you start getting really concerned about, well, all that is being then put into FishBase, and then we're using things like from FishBase to build these surrogate relationships or other things, and you start being like, well, have we built kind of this house of cards here? And we're hoping that we can kind of build some tools that help us solve some of those issues, and by bringing in other data sets. But that's certainly something we're thinking about. Yeah, Andre.
Yeah, the size selective thing, that's nothing short of collecting the right data. You can't really get around that from the sort of outside the assessment approach. So that is definitely an issue, a reason why we try to estimate growth internally, because you can account for selectivity because you're estimating it.
Yep.
There are cases, I'm thinking the southern shark fishery. I don't know to what extent you've worked with, I guess Terry Walker retired before you went to grad school, but we were able to estimate selectivity for things like gill nets outside the assessment, and then we built that into the estimator. So the actual likelihood function includes the size selectivity, but unless you've got size selectivity like that, those crazy negative T0s are just, that's what you get.
Yeah, for sure. The one advantage is if with these with the L naught version, if you can bring in any data on L naught, it can give you that anchor point. So you can at least get around the size selective bias against young fish. So if you're missing those really young fish, you can at least give yourself an anchor point. You're still going to have some bias that results in your Brody growth coefficient, just because you're going to have that selection for faster growing individuals at size, but it at least doesn't result in these just absolutely crazy T naughts that can result out of the back end.
Looks like we've run out of energy. It is the end of the call.
Well, I'm in summer semester, man.
We're not. We're still got a week of then exams and all that good stuff.
Absolutely.
Oh, you got Trinity again.
I was just going to say, if we have no more questions, we can start wrapping up. Thank you again, Zach, for presenting.
Yeah, absolutely. Thanks so much for the opportunity.
Yeah.
And I'd like to thank Trinity for all her work this quarter, for all the people who are online. I don't know how you do the whole clap thing. Apparently, Terrence can, but there's a clap. Thanks, Trinity, for everything you've done to make this all happen so successfully. And I'm going to thank in advance the lucky person who gets to do it next academic year.
Thank you, everyone, for coming. As Andres said, this is the last think tank of the year. We hope to see you again in the fall.
Yeah, thanks so much. Have a good one, everyone.